Hey there, we are in lesson 15, the human genome. We're going to talk about some human chromosomes, genetic disorders, and ways that we study the human genome. So, first of all, genetic disorders are diseases that you don't go catch these. The transmission isn't something that's contagious. It's something that's passed on from parents to offspring. And there's a couple reasons this could happen. It could be because parents are a carrier who pass this gene on for a disorder, or it could be because there's an error that occurs during gamete formation, or the formation of your sex cells. Remember, we talked about Patau syndrome, also called trisomy 13, back in Unit 5 when we were talking about cell division. We said, what are the differences between diseases that occur because of meiosis, errors in meiosis versus errors in mitosis? And since mitosis is only passing information onto yourself, your own cells are making other identical cells. Mitosis isn't going to be getting those problems passed on to your offspring. Whereas meiosis, you could see these problems being passed on to offspring. So let's talk about a couple things that could go wrong in gamete formation that could be passed on. So, first of all, let's look at mutations. We discussed the rules for base pairing. Cytosine has to go with guanine, thymine has to pair up with adenine. If there's a mistake, okay, the cytosine pairs up with an adenine instead. That's a mutation. And for the most part, our body does a really amazing process of repairing these mistakes before they move forward in the cell cycle. And there's a variety of enzymes involved in that, and that all takes place during the G2 stage of the cell cycle. Mutations can occur spontaneously, just a can't really explain it error in the DNA replication, or they can occur because of a mutagen, like what happened with the Ninja Turtles. So a mutagen is any agent such as a chemical, ultraviolet light, or radioactive element that can induce or increase the frequency of a mutation in an organism. Some mutations are harmless, like you don't even notice them. Your body just goes right on replicating, and there's no impact on the phenotype. Some mutations are beneficial. You see organisms that are actually better able to survive in their environment. And we're going to talk more about what happens there when we get into Unit 8 on evolution. Or some mutations can be harmful. And when you think about a harmful mutation, I always think about like an albino tortoise. If you're an albino tortoise, you're very light in color, so you're going to be easy for predators to pick out. And so that would be a harmful mutation. There's different types of mutations. Point mutations or substitutions cause replacement of single base nucleotides being replaced with another nucleotide. In a missense mutation, they're going to code for a different amino acid. In a non-sense mutation, they're going to cause that protein to be really short because it's going to code for a stop. And then in a silent mutation, it ends up coding for the same amino acid. And even though the DNA strand doesn't read properly, there is no visible impact to the phenotype because it still ends up coding for the same amino acid. So the protein structure will still be the same. Here you see a mutation where instead of T pairing up with A, T pairs up with C, and that's a point mutation. Here's an image of a point mutation where it was supposed to be a G, it ends up being a C, and that causes a stop codon to take the place, and it ends up shortening the amino acid chain. Examples of point mutations that occur, you can see in sickle cell anemia. It causes some red blood cells to have a sickle shape. You can look that up if you want to learn more about sickle cell anemia. Other types of mutations are called frame shift mutations. So whereas a point mutation just takes place at one point, a frame shift mutation shifts the entire sequence out of order. And I think the best example of that is when you read a sentence. If I just took out the letter T here, you could still read fat cat sat. So you know that the sentence is, you still know that the sentence is about a fat cat sitting. But if we shift this whole sequence of letters and you read the sentence as hef at, like you don't really understand any of that. Like that doesn't give you any information. So it kind of screws up the whole sequence. And that's what a frame shift mutation can do. We also have chromosomal inversions where the sequencing has kind of flipped. In this situation, you see it's supposed to be red, green, blue, orange, and the mutation flipped the blue and the green sequence. So that's where the error is there. Here's another image showing you how a frame shift mutation works. Luckily, when it comes to mutations, there are sequences of enzymes that are in the G2. They come out in the G2 stage of the cell cycle, and they play a really important role in repairing these errors before the cell continues forward through division. Sometimes they slip through though, and that's what happens. We also have genetic disorders where entire chromosomes are added or deleted, resulting in a genetic disorder such as trisomy 21, also called Down syndrome. Chromosomal errors occur because of non-disjunction. It's the failure of chromosomes to separate properly in meiosis. And you see the meiosis, and you should think of sex, S-E-X, sex cells form from meiosis. Gametes with extra or too few chromosomes result. Remember anaphase back in Unit 5 when we talked about the breakup, the anaphase. The failure of those chromosomes to separate during anaphase results in non-disjunction. And when you see the word junction, that's a point of two things coming together, like a train junction, two trains are coming together, or a junction box in like electric cables. Disjunction is breaking apart, and non-disjunction is the failure of something to break apart. So it's kind of a fun word to think about. A karyotype is an informative arranged picture of chromosomes at their most condensed state. We talked about this before. These chromosomes were likely collected during metaphase of mitosis. That's where they're most organized. They're lined up along the equator of the cell. You can see that there's 23 chromosomes in this human karyotype, and XY, you're a guy. So genetically, this is a male karyotype. An error can be discovered when we look at the karyotype and we say, oh, this guy has 3 chromosomes at the 21st location. So we would call this trisomy 21. And that's commonly known of as Down syndrome. And you're welcome to research these on your own to get a little bit more information. A third chromosome at location number 18 is called Edwards syndrome. Patau syndrome, which we spoke of, is a third on chromosome number 13. And Klinefelter syndrome impacts your sex chromosomes, and it's an XXY representation by having an extra chromosome at the 23rd location. So if these individuals have 47 instead of 46 chromosomes, that means the other gamete only has 45. So you're supposed to have 46, but because of non-disjunction, 47 went in one direction and 45 went in the other. Another way you can have a genetic disease is by being a carrier. An example of a genetic disease that comes as a result of being a carrier is Tay-Sachs disease. It's a hereditary condition in which an enzyme deficiency leads to an accumulation of fat and nerve tissue. Tay-Sachs can result in mental disabilities, convulsion, blindness, and ultimately death, unfortunately. Now, the way we show a carrier, it's another way of saying that you are heterozygous. So thinking back to Unit 6 and being heterozygous. Big R, little r is the dad. So he's heterozygous for this, but he's not showing it in his phenotype because it's dominant and recessive alleles. Same thing with mom. She's got a dominant and kind of recessive allele. So this disease is not visible in her phenotype. But if you were to take a moment and draw out a Punnett square here, big R little r crossed with big R little r, you would find one potential offspring is big R, 50% chance of them having or heterozygous representation, just like they are. And with each cross, there's a 25% chance that they could have a baby with Tay Sachs disease. So that's what it means to have a genetic disorder that is passed through your lineage as a carrier of that disease. Pedigree charts are commonly used to show a record of the family of an individual, and it can show the transmission of hereditary conditions. In this pedigree, you see the transmission of cystic fibrosis. So Wendell and Constance don't have cystic fibrosis, but Connie's a carrier. Edward and Sarah don't have it, but Sarah is a carrier. It just so happened that Wendell and Connie's son, Charles, gets together with Edward and Sarah's daughter, Eva, and Chuck and Eva have 4 kids. 2 of them have cystic fibrosis because, unbeknownst to them, that at this point they are both carriers. So that's how a pedigree chart works. Here's a more complicated pedigree that shows the transmission of hemophilia through a family tree of royalty. And this is kind of an interesting story if you wanted to look that up. So there's that. Okay. So again, as we've gone through a lot of things in this course, there's a moral spectrum, and that's between you and your family to decide where you fall on that. But given the knowledge and the ability to do genetic testing and to determine, if your offspring potentially has a genetic disorder, with that ability and to ascertain that type of knowledge comes some very tough decisions. And with that technology also comes the question of the appropriateness or the ethical considerations when you consider altering genomes or designing organisms and modifying their genes. So it's kind of a like, we can, but should we, kind of a question. And that's not for me to help you decide. That's for you to think about. And so yeah, that's where we're going to stop. So for chapter 15, we're going to stop here. I do have, of course, our favorite sisters, the Amoeba sisters, talking a little bit more about mutations. And you're going to read chapter 15, complete the chapter 15 reading quiz, and work on your vocab. And if you're far ahead, go ahead and do a choice board activity. All right, take care. Thank you.